Hello, everyone, and welcome to the fourth episode of the ANGIE podcast, where we discuss issues around targeted drug delivery. The ANGIE podcast has recently organized a competition for the best PhD thesis related to targeted drug delivery. The award for the best thesis recognizes excellence in research at an early career stage and encourages young researchers to pursue further career in the field of targeted drug delivery. I'm really happy today to have Dr. Souhaila El Moukhtari here with me, the recipient of the Best Thesis Award for her PhD thesis titled Nanomedicines for the Treatment of Pediatric Neuroblastoma, to talk to us about her research and her future plans. Hello, Dr. El Moukhtari.
Hello, thank you for having me, and of course, thank you for giving me this award. I'm very honored for this award.
We were really happy among all the competitors. We found that your thesis was the one that provided the biggest part of excellence that we were looking for. So this is why we decided to give you the award and to invite you to be our guest today in our podcast. So to start with, would you like to tell us a few things about yourself and how you started your career?
Sure, of course. My name is Souhaila, as you just said. I'm a researcher. I'm from France, but I live in Spain right now. I'm a researcher in a small biotech company, also in the field of targeted drug delivery. I did my PhD at University of Navarra, which is also in Spain, in Pamplona, under the supervision of Maria Blanco and Patrick Couvreur. And my PhD thesis was about the development of nanomedicines for the treatment of pediatric neuroblastoma. Although during my thesis, I also worked in the different types of cancers and different types of nanomedicines as a collaborator in Maria Blanco's group, of course.
Mm-hmm. And can you briefly explain to us what pediatric neuroblastoma is and why it is such a challenge to treat this type of cancer?
Of course. Well, as I mentioned earlier, my thesis was focused on the treatment of pediatric neuroblastoma. And pediatric neuroblastoma, it's a pediatric tumor that generally develops in very young children that are around 2 years old or less. And this tumor, it affects mostly the peripheral nervous system, but also some types of organs, such as the adrenal gland, that has a common origin with the peripheral nervous system. And the challenges with this disease is that it's a very well spread tumor that is also genetically very heterogeneous, so it's very hard to target the correct genetic aberration. And also it affects very, very young children, so it's very hard to find a treatment that is both very aggressive against the tumor, but not aggressive enough to damage these young populations.
Okay, and what has inspired you to focus your research on nanomedicine for pediatric neuroblastoma?
Well, in the field of nanomedicine, I was very passionate about targeted drug delivery and nanomedicine when I was studying pharmacy back at university. I found it very interesting to find drug delivery systems that were able to decrease toxicities and side effects that all the drugs have, and to target them specifically to their target site. I found that area very interesting in the field of pharmacy. And about pediatric neuroblastoma, I think it was my PhD supervisor that inspired me to follow the field of pediatric cancer. She is indeed very, very passionate about treating pediatric tumors, and she showed me all the possibilities that we had with nanomedicine, and I found it very interesting. Yeah.
Okay, and let's focus on your thesis more. Can you summarize the key objectives of your thesis research?
Yeah, of course. So the objectives were to take two molecules that were etoposide, which is a classic chemotherapeutic agent, and then edelphosine, which is another molecule that is not marketed yet. So these two molecules, they have some drawbacks when you use them in terms of properties and also in terms of side effects. So the idea was to encapsulate them, to put them inside nano devices, and to test if these nanomedicines could be combined in order to have an enhanced effect against tumors, but also to see whether these nanomedicines were able to reduce the side effects related to both treatments.
Okay, so you chose lipid nano devices in order to deliver the drugs?
Yes, so we decided to go for lipid nanomedicines. So the advantages of using lipid nanomedicines instead of other type of nanoparticles, because there are many ways to make nanomedicines. You can use proteins, you can use inorganic materials, you can use metals, you can use almost any material that is kind of okay for humans to use. But the advantage of using lipid nanoparticles is that most of the time, this type of nano devices, they are made of biological lipids, which means that they are biodegradable, they are going to be generally considered as safe. And there are a lot of types of lipids that you can use that are already approved by the FDA or the European agency, which make it easier to have a translatable option for the future. And right now, if you look at the market, the lipid nanoparticles are really in vogue, and they are being used by many private companies, but also by researchers because of the reasons that I've been telling you.
Mm-hmm. And you said you chose two different drugs to deliver, etoposide and adelphosine. Why did you choose these specific drugs? And is their capacity improved using the lipid nanoparticles?
Yes, so on one hand, etoposide, it's a chemotherapeutic drug that is already used for the treatment of neuroblastoma. So we know that this drug kind of works when treating this type of cancer and has most of the time a kind of good effect. So that was one of the reasons why we chose these drugs. However, etoposide presents several disadvantages when used in pediatric populations. Children can develop secondary tumors when they grow up. It's extremely toxic. It's very hard to have a solution made of etoposide because its solubility in water is very low. So it presented good advantages to use it against neuroblastoma, but also some drawbacks that could benefit from the use of nanoparticles. And then regarding adelphosine, the group of Maria Blanco at University of Navarra has extensive experience with this drug. We have seen that adelphosine used in combination with classic chemotherapeutic agents can be very efficient against pediatric tumors. We have seen very good effects of adelphosine against osteosarcoma in metastatic models. We have seen very good results in mice. That's why we decided to go for this drug. And we also knew that the analog of adelphosine, perifosine, was in clinical trials for neuroblastoma, so we knew that it could go very far for the treatment of this disease.
Right. Can you briefly explain for the people that are hearing how the tumor microenvironment influences the progression and the treatment of neuroblastoma?
So yeah, the tumor microenvironment, it's kind of the area where the tumor grows. So the tumor microenvironment, it's made of cells, elements that are not necessarily cells. They can be proteins or fibers and so on. So it's kind of the small world where the tumor cells are growing. And this small area heavily affects how tumors are going to grow, migrate, and also respond to treatments. So in the specific case of neuroblastoma, we have seen that tumor microenvironment can be very limiting in terms of treatment response. We have seen that the hypoxia that the tumor microenvironment generates, but also the different cells that makes this tumor microenvironment can be decisive when a treatment is going to work or not. So we decided to assess how the tumor microenvironment could interfere with the treatments that we're proposing. I don't know if it's. I don't want to be too specific because I think it's a very large area to explore. We have just explored a little bit in this thesis, of course. I think there are many people that know a lot more about tumor microenvironments.
Yes, but I think that we do have the general idea on how it is different for the neuroblastoma. What were the main findings from your studies, either in vitro or in vivo, regarding the effectiveness and the safety of lipid nanomedicines?
Yes. So regarding the efficacy of the nanomedicines, we have seen in cells that there was a combination. When we combine the nanoparticles, there was some type of synergism, which means that the efficacy of both treatments was significantly increased when compared to the single administered treatments at a specific ratio. So we were really happy about that. Then we also saw that the nanomedicines were safe in non-cancerous cells, both in vitro, but also in a C. elegans model, which is a worm that is usually employed in research because it has very many advantages. So this was a collaboration with a group of Ana Laromen in the University of Barcelona. And so once we knew that the nanodevices were safe and efficient in small models, we decided to go to mice, and we administered the nanomedicines first for a pharmacokinetic and a biodistribution study, where we saw that the nanomedicines significantly increased the oral bioavailability of the drug when administered orally, which meant that we could administer our treatments by the oral route. So this was a major result in the thesis, because we realized that we had increased concentrations of the drug without having enhanced side effects, which meant that maybe we could administer these treatments in the future to these children by the oral route. So this was a major result in terms of patient comfort, reduction of side effects, and all of the potential applications for not only for neuroblastoma but also for other tumors. And finally, we tested in. Sorry, yes, you were saying.
Is this the main difference compared to the traditional intravenous methods that are used to administer the drugs? The major benefit, let's say.
It's one. I don't think it's a major benefit, because etoposide is already given orally to some patients. But when you give etoposide without nanomedicines orally to patients, you have a very variable concentration, because the absorption is not very efficient. So you may have an increased absorption or very low absorption within the same patient. So it's not very a good option for this drug. I don't think it's the major advantage of giving drugs by nanomedicines to give it orally, but I think it's one of the advantages. And I think it's a major thing to consider when you realize that it's so hard for some people to get to a hospital, when you see all the costs that are implied with hospitalizations. I think it's more of a healthcare perspective.
Okay. And you had some more findings, but I interrupted you before.
No, please, you can interrupt me whenever you want. I think it's more fun that way. So I was saying that in the last part of the thesis, we performed an efficacy study in mice, in a neuroblastoma model. And this time we didn't see any amelioration of the treatment when compared to the standard of care, which meant that we administered the treatments both orally and intravenously in the case of the control drugs. And there what we saw is that most importantly, there was an important reduction of the side effects while the efficacy was maintained. So the model was not so aggressive, so all the tumors responded to the treatments, either in the nanomedicines or in the free treatments. So we didn't see any big amelioration because I think the treatments were already very, very aggressive by themselves. But we saw that the side effects were significantly reduced. So we saw blood side effects that appear with the free treatments that disappear with the nanomedicines. We saw hepatic, so liver toxicities, but that didn't appear when you were administering the nanomedicine. So there was a clear benefit from administering the treatments by oral route in nanomedicines. Yeah.
Do you see any potential long-term benefits of using lipid nanomedicine for treating pediatric neuroblastoma patients?
Yes. I think that we have to look at this issue as a long-term benefit that is not necessarily something that is going to cure very aggressive types of neuroblastoma, because I think that for very aggressive neuroblastoma, you have to find new treatments with new molecules, with all the advances that we have in biotech companies. But I think that this could be very beneficial for the quality of life of pediatric patients, because if they can receive the chemotherapy treatment at home without having to go to the hospital so many times, where they can stay in school, they don't have that many side effects. If we think about our children's life, it's not all about the treatment. They have to grow up as healthy adults, and for that, I think it's important for them to have a normal life, and I think to consider less toxic treatment that can enable you to have a normal life, kind of. I think that's the potential long-term benefit. Yes.
Mm-hmm. Indeed, it is really important. How do you think your findings contribute to the broader field of pediatric cancer treatment?
I think that since there are not so many research articles for pediatric cancer using nanomedicines, I think that any type of research that considers pediatric cancer treatment using nanomedicines is vital, because if you look at the literature, there are not so many people when compared to normal cancer that evaluate nanomedicines. And I think that's to contributing to that field in a scientific way. I think it's always positive for them. But as I mentioned earlier, I think that giving less toxic treatment to pediatric patients is vital, because there are a lot of very interesting articles now that evaluate how the toxicities that pediatric patients received in their childhood affect them when they are adults. You can see very severe depression. You can see important side effects in terms of cardiac toxicities, secondary cancer. So I think that in the long term, giving less toxic treatments to pediatric patients in general, not only cancer, I mean also for rare disease, for everything, I think it's vital, yes. And I think that studying how the reduction of side effects is important through nanomedicine, I think it contributes greatly to the field.
Yeah, sure. I think we have touched upon the improvement of quality of life after using oral nanomedicine. Do you think that there are any economic implications for using oral nanomedicine for cancer treatments in terms of health costs, for example?
Yes, I think that's a major issue that often as researchers in the academia, we usually forget, because we are in our labs and we we don't have to think about it because it's not our work. If we are scientists, we are scientists. We have to focus on the science, of course. But it's important to consider that a treatment can be applied to our current economic situation. I mean, I know for sure both France and Spain very well, and I know that the healthcare systems here and in France, they are suffering, and we keep closing hospital beds. Physicians struggle to find the financiation for treatments. So yeah, I think that finding affordable treatments for our healthcare system is vital as a country, but also as a European Union, because I think that the struggles that are facing France and Spain, I think they are probably brought to the rest of the EU. Although I'm not an expert in that, but yeah, I think that's very important. And if we think about other countries where the healthcare systems are not as strong as they are here in the EU, finding affordable treatments for cancer patients, I think it's vital if we think of other less fortunate countries, of course.
Yes. Do you think that you talked about osteosarcoma before, that you did some research about this, and do you think that there are any other types of pediatric cancers that would benefit from similar nanomedicine approaches?
Yes, I think that it's applicable to other types of pediatric cancer. Certainly, it depends then on the response to each treatment of the molecule itself. So it has maybe with other molecules that are more specific to other types of tumor. I think that the technology is applicable in any type of cancer, honestly. Yeah.
And what are the next steps in translating your research findings into clinical practice? Are you thinking of diving into clinical trials in humans?
Well, we are very far away from that, of course. I mean, I'm not working in this project anymore because, well, as I mentioned, I'm working in a biotech company nowadays. But of course, I keep in touch with my lab every day, and what they are doing now is that they are trying to put a specific peptide around the nanoparticles, a peptide that targets the tumor microenvironment. I don't know if you remember earlier, we spoke about the tumor microenvironment and how it was very complicated in neuroblastoma treatment. Well, this peptide, what it does is that it targets the tumor microenvironment, so it has active targeting properties for the nanomedicines, but it also affects the stiffness of the tumor microenvironment. As I mentioned, there are some stiffness in neuroblastoma, and thanks to this peptide, they are going to loosen up the tumor microenvironment so nanoparticles and drugs can enter in a better way. So here we are also talking about a synergistic activity that I started to work on during my thesis, but now they have a PhD student that is focusing on this research, mm-hmm. And I think it's very good that the group has continued working on this and to follow the steps that we started with my thesis. Yes, I'm very happy, honestly.
Yes, it's nice to see that progress is being made into previous findings.
Yes, I think it's important.
Mm-hmm. So did you face any major challenges during your research, and what was the way you used to overcome them?
Well, I found difficulties, of course, in the research itself, because honestly, doing a PhD when you start is complicated to find the correct tools in your brain to process all you have to do, because you have to read and you have to be able to understand what you're doing, because otherwise it doesn't make any sense, of course. So yeah, I found a lot of difficulties to have an organized brain while I was working, to know when I had to do each thing and to organize my experiments. But that was just part of the process. I mean, I didn't find it horrible or anything. But I found a lot of difficulties when COVID came, because I started in the end of 2019, and then we had the lockdown a few months after I started. So that was very hard as a human being, because the lab was alone, you have social distancing, and when you're a young researcher, a first year PhD student, you really need more support, I guess. And that was very lonely, but it was lonely for everyone, I guess.
Yeah, and you also need the interaction and the exposure to other people's research in order to improve your skills
Yes.
in the lab and in research in general. And
Yes.
right now, you're working in industry, right?
It's not well. It's a private company. Yes, it's not a big pharma. It's a startup it's in Santiago de Compostela. It's a spin-off from a research group from University of Santiago de Compostela, from the group of Maria Jose Alonso. And yes, we also work with nanomedicine. So I didn't go very far in terms of knowledge, because as I told you, I'm very passionate about this. And we also work in the field of cancer. So yeah, it's kind of almost the same, but we are still in a preclinical area. We are developing different types of nanomedicines using antibodies, small molecules, also. I think everything.
This is really great to stay in the same field, but to see things from another point of view. How do you think your research was received by the scientific community, and do you think, what is the impact you hope it will have?
Well, I'm not sure about how it was received. I can tell you how it was received by the people that I knew. I think that, of course, it had some limitations. The thesis has all the works, but I think it was received well, because I always kept in mind the whole process. I always tried during my thesis to always have the correct controls, to always keep in mind how to go further, to always keep in mind the scalability of what I was doing, to always keep in mind that our results were also that our experiments could be achieved by someone else that was reading the project. I always have to try to keep the scientific results very clean and very reproducible, or at least that's what I wanted. And I think there are things that were well received by the people that read my work. And yes, and also I think that the subject was well received, because people really want to know more about how nanomedicines can help pediatric cancer. And I think that was something that was especially well received, the subject, and how we worked on it with a lot of collaborators, with people that were specialized in neuroblastoma from University of Valencia, with people that were specialized in animal models. We try to keep specialist people helping most of the time, and I think that was important for the quality of the work.
I also was very impressed, and I think when I read the thesis, I really think that it adds value to the existing knowledge, and it takes the research part one step further on pediatric cancers, especially in neuroblastoma. That was your major field of research, but I think that also this is a very good example. The techniques that you have used is a very good example for doing the same for other types of cancer. Let's go to some closing questions. What advice would you give to other researchers or other students that are interested in the field of nanomedicine for cancer treatment?
Well, first of all, if there are people that want to go to a PhD, first to take their time to choose the correct group and the correct PhD supervisor, because this is the most important thing for me. I mean, without my group and my PhD supervisor, I would not be able to do anything, and to have a good relationship with them, for me, it was the most important thing, because it's a constant work. It's a very close relationship with a small group of people during at least 3 to 4 years. So that's very important. And I also will tell them to keep in mind that it's not a race, it's a marathon. So you have to take your time. Sometimes it's okay to take a step back and to rethink what you were doing, and to take some time for yourself, and all of the things that we don't think when we are doing the thesis, but then when we look back, we realize how important it is. And also to always, when you don't know something, to tell it. I mean, it's something that I always say to the students. You don't have to be afraid not to know something. It's okay not to know something and to be able to learn it. I think that we are constantly learning as researchers, and we have to be open to learn. Is the most important thing, I think, in this work. Yeah.
Great. And how do you see the future of pediatric cancer treatment evolving over the next years, over the next 10 years, for example, now that you have taken some distance from your PhD? Because I remember when I finished my PhD, I was so stressed that I could not see clearly the future of my work. It took me a few months to take some distance from it in order to understand how this would impact the rest of the field, of the research field. So what do you think now of the future of pediatric cancer treatment?
Well, I think that now, with the new treatments that are starting to appear, not necessarily the ones that I used in my thesis, but we have been doing incredible things with mRNA nanoparticles in the field of infectious disease and cancer. I think that definitely something is going to happen in this area. I've seen very interesting clinical trials ongoing with mRNA nanoparticles for pediatric brain cancer. And I think that they are going to ameliorate the treatment perspectives of children, thanks to this area, and also thanks to the CRISPR technology that is also revolutionizing the medical field, and also the CAR T cell therapies, CAR-M cell therapies, that are also giving a lot of new opportunities for cancer patients. So there are many things that are going on right now. So I think that not only pediatric cancer treatment is going to evolve, I think that cancer is going to be taken into consideration differently.
Mm-hmm.
And I think it's going to be like the boom that we had a few years ago with antibody therapies, with monoclonal antibodies. I think that we are living a new revolution. And I don't want to give too many details, but also I think that diagnosis is going to be much better thanks to artificial intelligence. I think that this is going to completely change how we diagnose people. It's going to completely change research and everything. So I think that we're living in a very interesting area for medical research, and I think that the next decades are going to be very interesting. And I'm really happy to be part of it, sincerely.
Well, thank you so much for being here with us today. It was a real pleasure having you and discussing about your thesis and about targeted drug delivery in general. And for the people that are listening, thank you all for staying with us, and please stay tuned for the next episodes of the ANGIE podcast. Bye, everyone.
Thank you.